when we talk about the example with the encyclopedia and we talk about the library, we're talking about the big picture idea here is the central dogma. The central dogma. If you could summarize that in as simple as possible, what might you describe the central dogma as? What might you call that? Or maybe let's put it another way. What do we use the central dogma to do in a cell? What's its purpose? Yeah, to build proteins, right? Yep. So the goal of the central dogma is basically it's how the cell builds proteins. How the cell builds proteins. So in a cell, I need a lot of proteins. We talked about in the last lesson what proteins are important for. They do all kinds of things in my cell. They have all those different levels of structure. So my cell gets its directions on how to build the proteins from a molecule that's called what? What's the name of the molecule that has information about how to build proteins?
DNA?
Correct. Yep. So DNA stores the directions for how to build proteins. That's its job. Our goal here at the bottom of the central dogma is we build proteins. That's where we want to get to do specific jobs in the cell. So my goal is to go from my molecule that stores information, which is the DNA, to the proteins that I'm trying to use to do something in the cell. Do you remember there was something in the middle that we talked about that I have to use to go from DNA to get to a protein? What's that other molecule called?
RNA.
RNA, exactly. So RNA helps me to take the directions to build the protein. So the three parts of the central dogma are DNA, which is what I start with, that stores my directions. And then I've got RNA that's going to take those directions out of the nucleus, and I'm going to end up with proteins. So we kind of mentioned this already. DNA is found in which part of the cell? Where does your DNA live in a cell?
Nucleus?
Yeah, it's inside the nucleus. DNA lives in the nucleus. And we just mentioned this with RNA. The reason I have to build RNA is I want to try to get my information out of the nucleus where the DNA lives. So RNA starts in the nucleus. Where does RNA go to actually be used to, or what organelle actually builds the proteins? Where does RNA go and attach to?
Ribosome.
Yeah, to those ribosomes. So RNA starts in the nucleus and goes to ribosomes. That's how I'm going to build my proteins. And then obviously those proteins, like we talked about in office hours before, proteins are found inside organelles, they're found in the membrane, and they're found in the soup, in that cytosol stuff. So we've defined here so far in the central dogma the three main parts of it: the DNA that we start with, the RNA that we're going to use to copy our directions, and then finally those proteins that we're going to end up with. And we talked about where we find them. The analogy that we had in the lesson and you have in your notes is an analogy about the library and encyclopedias. So the library and encyclopedias. So you remember from the analogy, basically what we're doing is we're trying to get a specific set of information that's stored inside the encyclopedia. I don't care about everything. I just care about one topic. So for example, tornadoes, like we were talking about in the example. That's kind of similar to the idea of how when your body needs a protein, we don't want to send the instructions for every single protein in your body. We just want to send the instructions for the one that I actually care about. So we want, when we're at that library, just to identify the part of the encyclopedia we care about, we want to take that information with us out of the library and get it home so we can use it to write our book report. When you were working your way through this analogy, I know you said we had trouble figuring out what transcription was represented, but what were some of the parts of this analogy that you were able to identify? What could you relate to different parts of the analogy? Oops, I might have lost you.
Hear me?
Oh, there we go. I can hear you again.
I'm sorry. I put my because my kids was screaming. They was like fighting. I don't know.
Oh, believe me, I understand. Yep, I've got 2 at home too, so I totally understand.
But I think it's not working. I have to use my computer. Okay. Okay. The encyclopedia, it's DNA.
Absolutely. Yep. So my encyclopedias represent DNA because I've got all of that information, everything I could ever need. So absolutely, that represents my DNA. What else were we able to identify?
Okay. I was like confused. The copy machine and the copy that are made. Okay. The process for the copy machine is going to come first. Yes.
So I'm using the copy machine to get the information that I want, right? I'm making a copy of it. So using the copy machine would be most like which process? So copying just the information I want.
RNA. RNA.
Yeah, so we'll close to that. The copies I make are RNA. So what I build by using that copy machine, those specific papers that I can take out of the library with me, those would represent RNA. The process of using the copier, so what I used to make my copier is helping me to go from DNA in the encyclopedia to RNA. What's the name of the process that takes me from DNA to RNA? When we're in the central dogma here, there's 2 steps. So going from DNA to RNA is the process of
Okay, I'm not sure. It's the nucleus. I don't know if I'm saying right. I just like try to figure out that the RNA it's come from the come from the ribosomes to the nucleus to the ribosomes.
Well, so the question I'm asking right now is when I have DNA and I turn it into RNA, what is the name of that step? It's actually the step that you came in asking me about in our analogy here. When I turn DNA into RNA, that's called
transcription.
Yeah, transcription. Turning DNA into RNA, that's the process called transcription. When I turn my RNA into proteins, that step is the process of translation. So remember when you were reading about the central dogma, there were 2 steps. There was the first step called transcription and a second step called translation. So taking DNA, all the information that I have in my nucleus, and turning it into RNA that can leave the nucleus, that process is called transcription. When I take that RNA, that specific set of information, and I turn it into proteins, and I do that outside, right, at the ribosomes, like you're talking about, that's the process of translation, turning it from RNA into proteins. So up here, we figured out that our encyclopedias are representing DNA, all the information I could ever need inside my cell. We've said that when we make copies, those copies that we make are kind of like RNA because we're only copying the information that we actually want. So when we're talking about the process of making copies, which is going from DNA to RNA, what did we say over on the left side of the screen there? What did we say that's called? When I go from DNA to RNA, that's called?
Transcription.
Exactly. Yep, transcription. The process of making copies is transcription. So I start with all the information I could possibly ever need in that encyclopedia, my DNA. I don't want all of it. I just want particular copies. There's particular pages I care about. That's my RNA. To get those copies made, I do the process of transcription, or I use my copy machine. Now, that was another one of those things that we needed to figure out in our analogy what it represented. So the copier, which is the machine, or if we think about it in terms of a cell, we could call it the enzyme that helps me to make my copies, or in my cell that helps me to build RNA. What is the name of that enzyme that helps me to build RNA? Do you happen to remember that?
RNA polymerase.
Exactly. Yep. So the copier is RNA polymerase. Its job is to build me RNA, or in my example that we're using here, my RNA is being represented by the copies. So the thing that helps me to build my copies is the copy machine. If I was inside my cell, it'd be RNA polymerase. Now, my encyclopedias live in the library. They can't leave the library, right? Just like the DNA in your cell can't leave which part of the cell? Where's the DNA in your cell found?
The nucleus.
In the nucleus, exactly. So the library is the nucleus. It's the place where I store my information. I store my encyclopedias, and I can't take those encyclopedias out. They got to stay there. So the nucleus of my cell in this analogy is represented by the library. Was that, I think that was all the parts of the analogy, right? Were there any other parts that we had to identify?
No, only this part. Okay, you have another part that's transcription and translation, and I have to compare and contrast it. And I didn't finish it because I was a little confused. I was confused also. The only part I saw. I'm clear with the information you gave to me. Thank you.
Okay.
Now I also understand it better.
Yeah.
And this one I have the nucleic acid, and it says the nucleic acid this process starts with, and then I have the transcription and translation.
Okay, so you want to work on that table from your notes?
Yes, please.
Okay.
I'm looking at that. And
let me take a picture really fast of this one just so I have it, and then we can clear a board and I'll put that table in for us. Okay. Let me see if I can find that table really fast. This is lesson number 6, correct? I think.
Let me double check. Yes. Yes.
Okay.
Number page 11.
Okay, so is this the table we were talking about?
Exactly.
Perfect. Okay. So let's see what we can fill in here already. When I am talking about the process of transcription, let's make a little note for ourselves. When I do the process of transcription, first thing I want us to remember, what do I start with and what do I end with? So remember on the page we were looking at before, we had the central dogma, which is 2 parts. The first part is transcription. So when I do transcription, I start with what? Do you remember?
DNA.
Start with DNA, absolutely, okay? And I end with what? What do I make at the end of it? RNA, exactly, end with RNA, okay? When I go over here to translation then, next door neighbor here, translation starts with what?
Translation starts with RNA.
Exactly, starts with RNA, and I end with, what do I build at the end of this?
Proteins.
Proteins, exactly. Okay, so let's look at our table. Now that we're remembering what we start with and what we end with, let's go through and look at our table here and see if we can figure out what some of our blanks are. So the first thing that we have on our table is it's asking us about the specific nucleic acids that we start with. Remember, nucleic acids are things that end with NAs in their name. That's what nucleic acid stands for. So with transcription, we said that we start with DNA right here, right? So the nucleic acid that I start with is DNA. We said that in its name. So the nucleic acid that transcription starts with, the kind of information basically that I start with in transcription is DNA. When we talk about translation, we said we start with the molecule called RNA, the nucleic acid or the information molecule called RNA. So translation starts with specific directions to make the particular protein that I care about. Transcription starts with DNA, which is all of the possible information that I could need in my cell. Now let's bounce down to our third part down here, excuse me, that asks about where these processes are happening inside the cell. When I talk about the process of transcription, do you remember where does transcription happen inside the cell?
You mean each place organelle?
Well, so when we talk about transcription, this process happens in the location in the cell where my DNA lives.
Okay, yes, this nucleus.
Exactly. Yep, transcription happens in the nucleus. That's the organelle, if you will, that transcription happens in. Now, once I finish transcription and I've made RNA, RNA leaves the nucleus and it goes out and it finds which organelle? Which organelle does translation?
The ribosome.
Exactly. So at the ribosomes. The ribosomes do translation, and they're the location of translation. And the process of transcription, since I start with DNA, DNA can only ever be in the nucleus. That's where transcription happens. Now, when I talk about transcription, and we're over here talking about the molecule that I build in this process, that's what we talked about up here, what I ended with. So I guess we could have filled in that column before, right? So I start with DNA, and the process of transcription ends with what? What did I say I built? So I'll give us a hint. We actually put it here for ourselves. The process of transcription, we start with DNA, and we build the molecule RNA. That's our goal at the end of this process. We use RNA. That's what we built.
RNA.
Yeah. So for transcription, we still built another nucleic acid. It still is an information molecule. When we get over here to translation, though, we kind of already filled this in our box above too. At the end of translation, we've actually built proteins. So proteins aren't an information molecule anymore. Everything with transcription, we're information molecules, right? But when we do the process of translation, we end up with a functional protein, something that does a job in the cell. So for the process of transcription, we started with DNA, we ended with RNA, and we did this inside the nucleus. When we talk about the process of translation, we started with RNA, we ended up building proteins, and the place in the cell that I do this is at those organelles called ribosomes. Does that make sense so far?
Yes, it does.
Perfect. Okay, so now we're down to our last part of the table here, where we need to figure out what each of these processes use. So when we're talking about the process of transcription, let's start on the left here with transcription. One of the things that we said that we built in the thing we said we built in transcription was RNA, right? There are two kinds of RNA that are listed over here, but I only make one of those kinds of RNA in transcription. Do you remember which kind of RNA I made in transcription?
T.
So T we actually use in the process of translation. mRNA is actually the kind that we build in transcription. mRNA, its M stands for messenger. So a good way to think about mRNA is this is the messenger that can go from the nucleus out into the cytoplasm where it can meet up with ribosomes. So mRNA, look, I guess we should make that more specific here. Let's add a little M in the front here. We make mRNA in the process of transcription. tRNA is something that I'm actually going to use in the process of translation. When we talk about tRNA, part of it had nucleotides, so part of it had letters, but there was another thing that was attached to tRNA. Do you remember what was found at the other end of a tRNA molecule? It got letters on one side, and what's the thing it has on the other side?
No, I need to check my notes. For the T. Okay, the amino acid? Or...
Exactly. Yep. Yeah. So on one side of a tRNA molecule, it's got an amino acid. And on the other side of a tRNA molecule, it has letters. So when you look at the picture of tRNA, like you found in your notes, it looks like the letter T with an amino acid on top and with letters down on the bottom on the other side. That's what tRNA looks like. When we talk about mRNA, it's just a string of letters. It's just a line. So the two kinds of RNAs, one of them, mRNA, is built in the process of transcription. The other one is going to help me with doing the process of translation. This is my translator molecule, is what I like to call tRNA, translator RNA. When we talk about what translation uses, the way that my tRNA molecules know if it's their turn to translate or if it's somebody else's turn to translate is because they look at the sequence of the mRNA. So notice here on our table how we said we started with RNA. I should probably add that extra letter, right? Let's make it this mRNA again. So we're going to make this back into mRNA. We started with mRNA. We used tRNA to help us to build proteins. That's kind of the sequence of events. So we start with mRNA, use tRNA to build proteins. You already told me that translation happens at the ribosomes in the cell, right? So we should also circle our ribosomes over here. The ribosomes are also involved in the process of translation. They're the ones who actually do translation. Let's go back to transcription because we didn't quite finish up transcription. In transcription, we built RNA, and you kind of already told me this when we were talking about the library analogy, but when we build RNA, when we use the copy machine to copy the directions we want, what's the name of that enzyme again that helps me to make my mRNA?
Polymerase.
Yeah, my polymerase down here. So RNA polymerase, that's going to be involved in the process of transcription. So it's building me mRNA. So far we've said that we build mRNA in this process. Here's the enzyme I use to build mRNA with. And the other thing that's involved in this process of transcription are the pieces that I use to build the mRNA. And those pieces are called nucleotides. So I string together my nucleotides, my A's, my U's, my G's, and my C's. I string them together using the enzyme RNA polymerase to build mRNA. So these are the things that are involved in the process of transcription. And these are the things, like we highlighted over here, that are involved in the process of translation. And what I'll mention for you, I'm going to put it here in a circle, a yellow circle too. I'm going to add that. The fact that mRNA, you can't even hardly see that, can you? The fact that mRNA is involved in both transcription and translation, that would be a good thing to know. I use mRNA in both transcription and translation. I can guarantee you'll probably have a question about that on the homework assignment and definitely on the test. Which process is mRNA involved in? Well, it's involved in transcription and translation. I use it for both of those processes because in one I build it, and in the other I use it as my direction to make those proteins.
Okay, I'm just wrote here, take a note. Okay. Okay, now it's much more clear to me. Even
Perfect.
like though I watch
Good.
the videos, know everything, and it's still, I feel, oh gosh, I think my head doesn't work as they used to be.
I can totally understand that. That is what I'm here for. So I'm glad that you stopped by and that we talked about these things because I can guarantee if you've got questions about it, I know you've got classmates that do too. So I'm glad that you stopped by for us to talk about it today.
Okay, thank you for your help.
You're welcome.
Bye-bye. Have a good day.
You too.